Throughout my studies, of course, I've been passionate about life sciences in general. But when I was doing my bachelor, a very unfortunate two events happened where I lost both my parents to antibiotic resistance due to co-infections. And because they were immunocompromised at the time, in one case, it was that the doctors did not have time to react with the right antibiotic. But in another case, which was more extended, which was more chronic, they just couldn't find anything else off the shelf that they could try. Unfortunately, I lost both my parents in 1 year after the other, 2010 and 2011. Since then, I've been really quite motivated to use my knowledge to go into personalized medicine, because I believe that in their cases, it's just that they couldn't find something specific to them, most importantly.
Antibiotic resistance kills 1.3 million people annually and could cost $1 trillion by 2050. Traditional antibiotics are failing against superbugs, leaving patients with few options. But what if artificial intelligence could revolutionize how we fight these deadly infections? Today's guest, José Bila, lost both parents to antibiotic-resistant infections, a tragedy that sparked his mission to develop AI-powered phage therapy solutions. His personal journey from chemistry PhD to biotech entrepreneur and now CEO of Precise Health reveals how cutting-edge science meets deeply personal purpose in the fight against antibiotic resistance. Welcome to the Smart Biotech Scientists. I'm David Brulman, your host. Join me today as José shares his journey from personal tragedy to pioneering AI solutions that could save millions from deadly bacterial infections. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brulman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, José, to the Smart Biotech Scientist. It's a pleasure to have you on today.
Thank you so much. It's a pleasure to be here.
José, share something that you believe about bioprocess development that most people disagree with.
I wouldn't say it's directly the bioprocess itself. It's more on the economics of the bioprocess, especially when you start thinking of more kind of personalized medicine, that usually people think it's a problem with scale up more than anything. But in reality, when it comes to the field in which we are, it's more about the economics regarding the batch utilizations. This is something that it's a little bit controversial, depending on who you speak to, of course. So for instance, in the case of a personalized medicine, you can have 1,000 liters of, let's say, of the product, but if you're only using for one patient, it doesn't change anything. You need to be able to have a high scale, say, batch utilization. These are things that can also be solved with different approaches like machine learning and so on. So it's not really a bioprocess per se, like I say, it's more of a general thing and regarding to the economics of bioprocessing.
José, draw us into your story. Can you share what sparked your initial interest in biotechnology and how your personal experience shaped your very scientific focus and led you to fight antibiotic resistance?
So I'm a chemist. I studied chemistry my entire life. I did my bachelor's, master's, and PhD in chemistry. And throughout my studies, of course, I've been passionate about life sciences in general. But when I was doing my bachelor, a very unfortunate two events happened where I lost both my parents to antibiotic resistance due to co-infections. And because they were immunocompromised at the time, in one case, it was that the doctors did not have time to react with the right antibiotic. But in another case, which was more extended, which was more chronic, they just couldn't find anything else off the shelf that they could try. Unfortunately, I lost both my parents in 1 year after the other, 2010 and 2011. Since then, I've been really quite motivated to use my knowledge to go into personalized medicine, because I believe that in their cases, it's just that they couldn't find something specific to them, most importantly. From there, after my master's, for instance, I worked as a chemist. Of course, I was designing molecules and synthesizing molecules for photodynamic therapy. So it's another, say, a strategy that is used for cancer, for instance. That was very deep personalized medicine. And after my PhD, I went into management consulting, specifically in life sciences. And there I learned a lot about gene therapies, personalized medicine, and so on. And all this knowledge and my personal story is what culminated into precision medicine, our startup that we have built today.
It's a very tragic story indeed. And at the same time, I love your purpose behind that and have a greater purpose and not just science. Well, it comes out of a personal experience, a tragic one, and now you're using that for the greater good, which is very powerful. So I am wondering, Jose, to what extent this tragic event is an isolated case, or can you tell us a bit what is the current state of antibiotic resistance? How many deaths do we have per year, and what is the costing to our healthcare system?
So estimations from World Health Organization and all the different surveillance programs we have estimate that there are 1.2 to 1.3 million deaths globally per year caused directly by antibiotic resistance, but there are actually around 5 million which are indirectly caused by antibiotic resistance. Of course, we usually look at the direct ones, but the indirect ones also, these co-infections that lead to something else that leads to something else and is reported not as a death by antibiotic resistant, but by organ failure or something else that we never learn about. So the number is huge. And then estimations again by 2050 indicate that there will be about 10 million deaths globally per year. So this is a huge number if you look at it, and it's quite comparable to cancer today. The number is increasing day by day, and some even newer estimates indicate even more than 10 million deaths today. This is a big problem. It's not only a question of the deaths themselves, there is also a cost burden which is associated to this, because when a patient has an antibiotic resistant infection, the tendency is that they will stay longer at the hospital and this patient is not going to work. You have more doctors having to focus on one specific patient and so on. So the economics globally actually show about 1 trillion additional cost by 2050 globally for the healthcare alone. So this is a huge number associated to the 10 million deaths that are expected. I think the bottom line is that we need to find a solution that works. The current antibiotics are failing, and this is not working as we have today. As you might know, and also your listeners might already know, a lot of the pharma companies are actually exiting the entire antibiotic space because the economics of it are not very, say, attractive compared to cancer and many other, let's say, indications that you could think of. Again, since 1950, I think it's been more than 50 years, if some reports show that there hasn't been a new antibiotic that has been very effective compared to what we already have today against gram-negative bacteria. Again, gram-negative bacteria is the one that is causing a lot more damage than maybe gram-positive. I mean, in relative terms speaking, of course. Finding new modalities is important. Finding new approaches is very important. This is why I think a lot of the innovation has to come from maybe smaller companies, because the big ones are exiting. We should have collaborative approach from different, let's say, institutions, academic, SMEs, and private, and so on. So it's a big problem, because what we don't consider as well is that out of these 10 million people that will be dying, it could be my parents that died, but it could be somebody else's parents, or could be us exactly that could be dying next. So this is a big problem for sure.
It definitely is a big problem, and the numbers are crazy. And antibiotic resistance has been a topic or hot topic for several decades already. So what is the current state now in 2025? Is it still the main drivers that we've talked about for the last decades, or is it something else that's causing even greater numbers of people that are probably going to die in the future?
One of the drivers, of course, is caused the antibiotic resistance in the first place. You can think of the antibiotics being these static molecules because you synthesize a chemical molecule and that's static. You're not going to change. I mean, I'm a chemist, you're also a chemist. So you know that even changing one substitution on one of the, let's say, positions of the molecule, that would require you going through again the clinical trials, which can take 10 to 15 years until you can actually commercialize it. So this static molecule is fighting against something that is changing. It's adapting itself. So if you are in a box fight and you keep throwing punches the same way to the same direction, if I am adaptable, of course, I will know after a few punches that I need to run away and I know how to defend myself against that. And that's the bacteria is actually doing. So we need an approach that is adaptable as the bacteria is also adapting. So that is one of the things. So having static approaches is one of the things that is causing or is increasing the issue with antibiotics. And the other thing is that, of course, over time, I mean, depending on which culture you come from or which country you come from, people, whenever they have a small symptom, the intuition is to go and get paracetamol, maybe, for example. But in some cases, most people are like, oh, just take an antibiotic. So the misuse and overuse of antibiotic has been driving the entire, let's say, issue with the antimicrobial resistance altogether. Again, on the adaptability aspect, on the pharma side, if you have to spend 10, 15 years to develop a new drug and spend billions of dollars, that then at the end, the economics of it makes sense, but the margins are very small, then this makes less attractive to actually find something that you can adapt toward this bacteria that it keeps changing. These are all things that are coming together, and we've known them for ages, actually, just that there isn't much being done in that space.
So how are we going to solve that problem as an industry, and more specifically, how are you solving that in your company?
So in our company, we are focusing on bacteriophages. So bacteriophages, they're viruses that are present where bacteria is present. And they're usually there to either control the growth or to eliminate pathogenic bacteria and in general. But what they do is that they infect and kill specific bacteria. And they're very specific and they're natural. You can find them from many, many different sources. And in our case, we get them, for instance, we isolate them from wastewater, or we isolate them from agricultural products, or we get samples from people, for instance, which are donated, and we isolate them. So they're very good because they are very specific to the strain level or a group of bacteria strains in these cases. They are harmless to the human cells, or at least that's what has been indicated by far. So they're quite safe compared to maybe chemical molecules, in this case, the antibiotics that we could be using that are not quite toxic, but could be toxic in certain instances. These bacteriophages can be used as a weapon against, let's say, bacteria that is pathogenic and is resistant against antibiotics. And that's what we are focusing on in our startup.
Before we continue, biotech founders, I want to talk directly to you. Do you know that feeling when an investor asks about your manufacturing strategy and you freeze? One wrong CMC decision can cost you $2 million and 18 plus months of delays. Yet many CEOs are flying blind on these critical decisions. That's why I created the 1-week CMC strategy assessment. In 1 week, you get a personalized assessment of your molecule and development stage. We identify critical gaps before they become million-dollar mistakes, plus a strategic roadmap for high-stakes investor meetings. One CEO told me, David took me from feeling powerless about our CMC roadmap to confidently pitching investors. An absolute game changer for time-pressed CEOs. Whether you are preparing for Series A, approaching IND filing, or facing scale-up challenges, this gives you confidence to ace investor and board conversations instead of surviving them. Ready to stop second-guessing your CMC strategy? check the link in the show notes and secure your spot. Because your breakthrough therapy deserves a solid CMC strategy. Now, let's get back to our episode. And I imagine these bacteriophages, they are very specific. So you have to do some analysis to know which one you have to pick. And then how do you find the needle in the haystack?
Exactly. So bacteriophages have been with us for over 100 years and have been actually used in many different locations, specifically ex-USSR geographies have been using them. Currently, Georgia has been using phages for quite some time. But the issue that makes them maybe less attractive compared to antibiotics, because antibiotics, like I said, is this chemical molecule that you can give to a bunch of people. If they have the infection, it's going to kill all the infections, different infections, but it happens that maybe a certain percentage it's not able to kill, right? But phages are too specific, and being too specific means that you have to personalize to the specific individual. So this makes the economics a little bit difficult in different levels. And then back to your question regarding on how we find the right one. Today, in order to find the right one, essentially you need to take the bacteria isolate and then look within a specific library of bacteriophages and test one by one manually in order to find the right one. And you do this using plaque assays or even different strategies like liquid assays and so on. So you can essentially find the right phages, but it's done currently manually. And this process is laborious and is one of the reasons a lot of the doctors are not using. So for instance, we talked to different doctors since we're in this space. We just wanted to understand, we just posed a simple question. Bacteriophages are specific, they are amazing. They can even penetrate biofilms, which is one of the key issues with antibiotic resistance. And they have less toxicity compared to existing maybe antibiotics in general. So why aren't you using them more? Even when you have a patient that is dying, why not resort to bacteriophages? And the key issue that most medical doctors pointed out was that in order for them to find the bacteriophage, it can take anywhere from 2 to 6 months. And this is insane. Imagine a patient that has, say, an acute or subacute infection, which is a good portion of the infections that we have today. So you don't have time. You need to react fast. Normally, if you isolate the bacteria and then you have to ship it to, let's say, a phage bank that is somewhere, maybe you're in Switzerland and this phage bank is in Georgia and then another one in Portugal, you have to accept the fact that even if you ship it, they might not have what you're looking for. So the entire manual process and so on, and then the production, etc. It's a whole nightmare. And this is where we think machine learning actually can add something, because using machine learning approaches, what we can do is to use specific traits of the bacteria and specific traits of phages, and using that combination, we can learn by looking at what works and what doesn't work, and understanding what doesn't work, why it works or why it doesn't work, and train a model which can specifically identify the right phages or specific bacteria strain without having to go through the entire hassle. And this is what we are doing at Precise Health, which is our startup. And here what we are doing is developing this machine learning platform. We're interconnecting multiple phage banks, and what the doctor would have to do instead of doing the manual processing and shipping of bacteria, etc., all they need to do is to have a DNA information of the bacteria and some metadata regarding the bacteria and upload that in the platform. And we scan through multiple phage banks at once, and this is done within minutes. And we can already know which phage bank has which bacteriophage, and is the bacteriophage already ready to be used in a patient under, let's say, the local regulations in this case. So these are things that we can do using and machine learning in general.
So we have the traditional approach that takes weeks, if not months, to find the right bacteriophage, and by then probably the patient will be dead or in a very bad condition if everything goes well.
Yeah.
Now, with your machine learning approach, what is the potential of that? How fast will that identify and then produce also that bacteriophage?
The big dream for us is to have multiple phage banks holding already produced and qualified phage, let's say batches, so to speak. And in that big dream, the idea would be that when the doctor already decides that, oh, my antibiogram shows that nothing in the shelf that I have is going to work, then can I use bacteriophages? And usually this can be done anywhere between 48 to 72 hours, of course, depending on the hospital capability. It really depends on which country you are. But in Switzerland, we know that this can be done by hour 72 for sure. And with that in mind, can I just search within minutes and get my phage? I don't know, even if it's somewhere in Finland, can I just in 2 clicks request the phage, and that the Finnish biobank, which has the ready to use phage already produced, send that to me next day shipment, depending on the severity of the case that I have. So our target actually is by day 5, you get your phages in-house to be used under whatever regulations you're following in your specific country. So that's the idea. But there is another issue. The production itself is an issue because, isolating phages is not the issue. We have more than 200 phages in our laboratory, so we can go and produce all of them. But then it comes back to my very first comment regarding the bioprocessing. If I produce all the 200 phages in whatever batches, how likely am I to use a certain percentage that is going to make the economics actually right? So that is the key question. Again, coming back to our machine learning algorithm, what we can do is to actually, by looking at specific, let's say, regional fingerprints, we can already anticipate which phages within certain phage libraries or phage banks can maximize the chance of being utilized. Just a simple example. If I go to a hospital in Switzerland and let's say in Zurich, we often have kind of the same bacteria patterns that are running around because the population is the same. They are eating the same thing. They are breathing the same air, whatever. The travel patterns are quite, let's say, relevant. So by looking historically at the different bacteria that has been, let's say, deposited and isolated in the banks of the hospitals, can we use that information to sort of predict or develop a fingerprint for the region of Zurich in this case? And then with that fingerprint, can we look into the phage libraries that we have incorporated in our machine learning platform to scan for the phages that are likely to cover 80 to 90% of the fingerprint for Zurich? And can you do this for multiple cities? Maybe it's not Zurich, maybe it's entire Switzerland, or I don't know, entire Europe. We don't know for sure for now, right? But that's what we are working towards to understand. And by doing so, let's say for Zurich, you need X amount of phages that are likely to cover 80 to 90%. Can you already take those and get them pre-produced and stock? And when the doctor needs them, they're already produced because you already know that it's very likely that it's going to be one of those. And then in cases where the 10% is not covered, then you can come back and search again within the libraries. And of course, if the patient still has time to cope with the infection, then this would have to be taken into, again, production, etc., and be taken into the library. And then using the exact same system, what if you were to get a continuous bacteria surveillance, let's say, program from this hospital that I'm talking about in Zurich? Every, 3 to 6 months, we collect a new bacteria and we check again, are we still covering 80 to 90%? Or are we down to, 70 or 60%? And then can we scan again and say, how do we get back to the 90% and do the production? And then you have a continuous surveillance program and you have a continuous update of the bacteriophage which are produced. So this is the way we believe is to go, and you reduce time, you make it fast and more effective and let's say more targeted, and then the economics will be correct.
That's a wrap on part 1. José's story shows how personal tragedy can fuel scientific innovation. In part 2, we'll dive deeper into the economics and regulatory challenges of AI-guided phage therapy. If you have process development or manufacturing questions, book a free call at brumanconsulting.com/call, and I'm happy to help you get started. And also, please leave us a review on Apple Podcasts or whatever platform you found us on. It helps other biotech scientists discover actionable insights like these. Thank you so much for tuning in, and I'll see you next time. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.